Imagine taking three completely different natural disasters, like a hurricane, a wildfire, and a massive earthquake.
Right, completely different forces of nature.
Exactly. They tear through a city, and when you go to inspect the wreckage, you discover something that, well, it should be impossible.
Oh, this is fascinating.
Every single one of them left behind the exact same incredibly specific, perfectly shaped footprint in the rubble.
Which, logically makes no sense. They have totally different mechanics.
Right, but that is exactly what scientists have just found happening inside the human brain, and that's what we are getting into on today's deep dive.
It really is a massive biological aha moment. It completely upends how we traditionally categorize brain disease.
So our mission today is to unpack this really groundbreaking 2026 paper from the journal Cell by Zhu and colleagues.
Yeah, and we are going to connect a lot of dots here. We're looking at how three major, fundamentally different neurodegenerative diseases actually share a hidden mechanism of DNA destruction.
Okay, let's unpack this step by step for you, because we are going to get into some dense biology, but we'll explain every piece of jargon.
Definitely. Today is all about the molecular why, not just the what.
So let's lay out the suspects first. On the surface, these diseases are about as different as our hurricane and earthquake. We've got ALS and frontotemporal dementia, or FTD, on one side.
Right. And in medicine, we usually put ALS and FTD in the same box. They both involve problems with a very specific protein called TDP-43.
And normally TDP-43 is a good thing, right? It lives in the nucleus of the cell.
Yeah, it carefully tends to the DNA. But in ALS and FTD, it goes rogue. It basically vanishes from the nucleus and glumps up in the surrounding fluid, the cytoplasm.
And I know this is often linked to a genetic mutation, right? Like a stutter in the DNA code.
Exactly. It's called a C9orf72 repeat expansion. Just a nasty little genetic glitch.
Okay, so that's ALS and FTD. But then you have Alzheimer's disease,
which operates in a totally different neighborhood of pathology. I mean,
Yeah.
you don't see that TDP-43 problem there.
Right. In Alzheimer's, you hear about amyloid beta plaques outside the cells and those tangles of a different protein, tau, inside the cells.
Right. Completely different genetic risk factors, totally distinct protein clumps, very different symptoms for the patient.
Different weapons, different crime scenes. So to figure out what these diseases actually do, the researchers decided to look at the DNA of individual highly diseased brain cells.
Which, frankly, sounds like science fiction.
I mean, how do you even do that? You have billions of cells in the brain. How do you isolate a single diseased neuron's DNA without just destroying the evidence?
The technical gymnastics required are staggering. They relied on a technique called FANS.
SANS, what does that stand for?
Fluorescence-activated nuclear sorting. You basically have to turn the brain tissue into a cellular soup first.
Oh, wow. Soup.
Yeah. And then they use custom antibodies that attach to specific targets, like a missing TDP-43. And these antibodies literally glow under a laser.
So the machine just looks for the glowing ones.
Exactly. They fire a laser at the soup, and the machine sorts out only the specific diseased neuronal nuclei they want.
But even then, one cell only has 2 copies of its genome, right? That's almost no DNA at all.
It's a vanishingly small amount. If you just try to sequence that directly, you'd miss most of it. So they use something called PTA.
And what's PTA?
Primary template directed amplification. It carefully, almost flawlessly, copies that tiny DNA trace thousands of times.
Oh, so they amplify it first.
Right. And only then can you run it through SCWGS, which is single-cell whole genome sequencing. They managed to read 469 individual neurons this way.
That is wild. And when the data came back, the scale of the damage they saw was just terrifying.
Yeah, they found that neurons from all 3 of these diseases showed a massive, accelerated accumulation of somatic mutations.
Meaning mutations happening in the actual living cells of the body, not inherited ones.
Exactly. And they were looking at 2 specific types,
Yep.
SSNVs and SINDLs.
Okay, let's break that down. I always picture the genome as like the ultimate instruction manual. So SSNVs, single nucleotide variants, those are like typos, right?
Yeah, exactly like typos.
Yeah.
Instead of typing bear, the cell types pair. It might be harmless, or it might change a sentence.
But the SINDLs, the insertions or deletions, those are much worse.
Way worse. And SINDLs like pouring glue between pages of the manual or violently ripping a whole paragraph out.
The code just shifts. Everything after that ripped page becomes gibberish.
And the sheer volume of those ripped pages was shocking. In a healthy aging brain, you might get a few dozen SINDLs over 80 years.
But in these diseased cells,
They found cells with over 1,000 SINDLs. It's the equivalent of hundreds of years of normal genetic damage packed into one lifespan.
The genome is effectively shattering.
Coding was shattering.
Okay, let's do a quick recap here for you listening and make sure we're all on the same page.
Good idea.
So, bullet 1. ALS and FTD, with their TDP43 issues, are categorically different from Alzheimer's and its tau pathology.
Right. Bullet 2. Using that crazy single-cell sorting and sequencing, scientists found massive accelerated DNA damage, those SNDLs and SSMVs, across all 3 diseases.
So here is a question for you to chew on. Think about a time you tried to read a corrupted computer file. If the brain's instruction manual is missing huge chunks of code, how does the cell even keep functioning? And what stands out to you about the fact that different diseases cause the same corrupted files?
A great question, because it really shouldn't happen that way.
Right. I'm getting stuck here. If TDP43 and tau are totally distinct disasters, why are they ripping up the manual in the exact same way? Like if I hit a computer with a hammer versus dropping it in water, the hard drive fails differently.
Yeah, and that intuition drove the researchers to look at the actual shape of the tear. They used computational extraction to look for mutational signatures.
Like tire tracks at a crime scene.
Exactly. DNA mutations leave recognizable patterns.
Yeah.
And they found one shared signature across all 3 diseases. They called it IDA.
IDA.
Yeah.
And it is astonishingly specific. We aren't just talking about random chunks of DNA going missing.
No, it is always exactly a 2 base pair deletion. 2 tiny rungs on the DNA ladder, consistently snipped out.
But it gets even more granular. It only happens to specific letters. The genome is written in A, C, G, and T. This IDA footprint specifically deletes a CT pair, a cytosine, and a thymine.
Right, but only in a very specific neighborhood. It deletes the CT only when it's sandwiched inside a sequence called TNT.
Thymine, any base, thymine.
Exactly.
So we have a simple mnemonic tool for you to remember this. Just think. CT gets cut in the TNT.
That's a great way to remember it. CT gets cut in the TNT. Out of 3 billion base pairs, the damage targets that exact microsequence.
And the prevalence of this is insane.
It really is. They found this IDA process actively destroying genomes in 22% of ALS neurons, 76% of FTD neurons, and 61% of Alzheimer's neurons.
And what about the healthy control neurons from people without the diseases?
Barely 2%.
Wow. So this footprint, the CT cut in the TNT, is practically the defining feature of these sick brains.
Yeah, it's essentially absent in healthy tissue.
Okay, time for our second mini recap.
Let's do it.
Bullet 1. The DNA damage isn't random. It matches a mutational signature called IDA.
Bullet 2. IDA is defined by 2 base pair deletions, specifically losing a CT inside a TNT sequence.
And bullet 3. This footprint is everywhere in diseased brains, but almost completely missing in healthy ones.
So think about this for a second. If you were a molecular detective and you found the exact same CT cut in the TNT footprint in totally different neighborhoods of the brain, who would you start looking for?
What biological machine targets that specific sequence?
Right. The footprint leads directly to one very specific suspect, an enzyme called topoisomerase 1, or TOP1.
TOP1. Okay, let's unpack TOP1. To understand why it's cutting the DNA, we need to know its day job.
Yeah, normally it's a biological hero.
Imagine DNA as one of those old-school twisted telephone cords. When the cell needs to read the DNA, motor plows between the strands, pulling them apart.
Right, a process called transcription. But as you force them apart, the rest of the cord supercoils. It winds tighter and tighter ahead of the motor.
And the physical tension is immense. The DNA backbone would literally snap if that tension wasn't relieved.
So TOP1 is the cell's elegant solution. It acts like a highly specialized pair of molecular scissors.
It clamps on, snips just one strand, lets it swivel to release the tension, and then...
crucially, it perfectly tapes it back together, millions of times a day.
So how does our hero become the villain?
Well, it requires a perfect storm. Step 1 is the environment. Neurons use a ton of energy, which creates massive oxidative stress.
Basically the biological equivalent of rust.
Exactly.
Yeah.
And that rust damages the DNA bases, forming toxic variants like 8-oxidase.
Okay, so the DNA is getting rusty.
Right. Step 2. Because of this toxic environment, the machinery that copies DNA gets sloppy. It accidentally grabs the wrong pieces called ribonucleotides or RNTPs.
Those are RNA building blocks, right?
Yeah, and it glues them right into the DNA helix.
You can't have RNA glued into a DNA strand. It ruins the structural integrity.
Exactly. So normally an enzyme called RNase H2 acts as a cleanup crew. patrolling for this junk and removing it.
But let me guess, in these diseases, the cleanup crew is missing.
You nailed it. RNase H2 is massively downregulated in ALS, FTD, and Alzheimer's.
So the RNA junk just builds up.
And that leads to step 3. TOP1 sees this mess and tries to step in to do an emergency repair job called RER.
Ribonucleotide excision repair.
Right, it's a desperation move. TOP1 goes in and cuts the DNA to remove the RNA piece, but it hits that oxidative rust, that 8-oxyG.
Oh, no.
And it gets physically stuck. The molecular scissors cut the DNA, but then get welded to the strand. This creates a highly toxic lesion called a TOP1CC.
A TOP1 cleavage complex. So the tension reliever is now glued to an open wound in the DNA.
Leaving the genome with an unresolved single-strand break, or SSB, the cell is panicking. It has to deploy an aggressive salvage pathway to literally chop out the trapped TOP1.
And because of how TOP1 binds to the DNA when the cell chops it out,
it mechanically, consistently chops out exactly 2 base pairs.
the CT gets cut in the TNT.
Exactly. The IDA signature is literally the scar left behind when the cell amputates the trapped POP1 enzyme.
That is mind-blowing. And they proved this mechanism with some serious biochemistry, right?
Oh, yeah. They used a radar assay, which confirmed a huge spike in TOP1 enzymes physically welded to the DNA in diseased brains.
And what about the meta-CS technique?
That's duplex sequencing. It reads both sides of the DNA ladder to prove that these started as single-strand cuts.
And then there were the gels.
Right. S1 nuclease gels. S1 aggressively hunts for single-strand breaks. In healthy DNA, the gel shows clean bands. But in diseased DNA...
It's just massive smears on the gel, right?
Yeah, proving the genome is just riddled with thousands of microscopic cuts.
Okay, let's do our third recap.
Go for it.
Bullet 1. TOP1 usually entangles DNA safely, but oxidative stress forces it into emergency repair, or RER.
Bullet 2. The oxidative rust traps TOP1 onto the DNA, leaving dangerous single-strand breaks.
Bullet 3. These breaks result in the 2BP deletions, the IDA signature, and genome fragmentation.
So here's where it gets really interesting for you. The brain is essentially destroying its own DNA, trying to fix it. How does knowing this change your view of neurodegeneration?
Is it a disease of attack or a disease of failed repair?
It's a huge perspective shift, but it also raises a big question. If neurons are doing this, is the whole brain doomed?
Right, which brings us to the control variable, the cerebellum.
The cerebellum is at the back of the skull, and crucially, in ALS, FTD, and Alzheimer's, it's largely spared.
Patients might lose memory or motor function, but the cerebellum survives.
So researchers checked the cerebellum neurons using the exact same FANS and sequencing techniques.
And what did they find?
A complete absence of the IDA signature. No smears on the gels. The catastrophic damage is highly region-specific to the cortex.
So it's not just a general artifact of a dying body, it's tied to the disease in those specific vulnerable neighborhoods.
Yes.
But there's a fascinating twist here for everyone listening. This IDA process isn't completely alien, it's actually a feature of normal aging, isn't it?
Yeah, as we age, we all accumulate a few of these CT cuts from normal oxidative stress.
Yeah.
Neurodegeneration is basically normal aging hitting hyperspeed.
Wow. Now, we should mention researchers still debate if TDP43 or tau is the primary driver of these diseases.
Right, we aren't taking a stance on the original trigger. But the beauty of this data is it shows that regardless of the trigger, the terminal pathway involves this TOP1 DNA shredding.
It's a unifying bottleneck of destruction.
Exactly.
Okay, final recap before we wrap up.
Bullet 1. The cerebellum is spared from this damage, proving its link to specific disease pathways.
Bullet 2. This 2BP dilution happens slowly in aging but goes into overdrive in neurodegenerative diseases.
Bullet 3. TOP1-mediated damage is a shared catastrophic endgame for totally different brain diseases.
So, if multiple different diseases share the exact same endgame, should future medicines target the original causes or target this final bottleneck?
That is the million-dollar question. For decades, we've tried to clear amyloid plaques or fix TDP43 with mixed results.
We keep trying to stop the hurricane or the earthquake.
But what if we just reinforce the buildings against that final footprint?
Right. We know inflammation creates the rust. If we can't stop the genetic triggers, maybe we can protect the brain by making sure TOP1 never gets stuck.
Could a treatment stabilize TOP1? Or better yet, boost RNase H2 levels so the normal cleanup crew comes back?
A universal shield against dementia and ALS, it completely reframes how we look at the aging brain.
It reminds us that even when diseases look totally different, the deepest mechanisms of life and death follow the same rules.
And if we can decode the shape of the tear, maybe we can finally learn how to stop the scissors. Keep questioning the world around you, and thanks for joining us on this deep dive.